I talked about why reporter constructs are useful a little bit in the previous lecture, and I just wanted to go over it again in a little more detail here. So let's say that you have pulled out a clone from your genomic library, and you you're interested in protein X. You used a probe for protein X, and you were able to find a clone that had the entire sequence of protein X and also the upstream regulatory elements. And you'd like to know, what is the expression pattern of this gene? I would like to know what tissues of the body is it expressed in. But it's not easy to answer that question, because you'd have to either, you know, test every single tissue, or you'd have to have an antibody to detect the protein in every single tissue, and that's kind of technically challenging. It's oftentimes easier to just create a reporter construct, which would be replacing the red gene for the green gene. The green gene is going to be something that you can see, something like a fluorescent protein, or oftentimes beta-galactosidase, which is an enzyme that creates a blue color when you give it the right substrate. And that's hooked up to the same regulatory elements as your original gene of interest. And then you can see, yeah, it's not expressed in tissue A, but it is expressed in tissue B. It's not expressed in C and D, but it is expressed in E and F. And then you can further test it to see what the contributions of these individual regulatory elements are. See what, if you only have element 3, what happens? If you only have element 2, well, it's expressed in these tissues. If you only have element 1 by itself, it's not expressed at all. But 1 and 2, it's expressed in E and F. So questions like this can be answered through reporter constructs. The reporter construct needs to be introduced into a living organism, or at least a living cell, to test it, and we'll talk about how that's done in a moment. Before I do that, I wanted to say a word about PCR, which is that DNA libraries used to be the only way to clone DNA. They used to be the only way to make many copies of DNA. And libraries are still used for very large genes, and they're still used for sequencing whole genomes. But for stretches of DNA that are up to about 10,000 base pairs, PCR is now the technique of choice. It means that you don't have to create a library. A library is no longer the first step for cloning a stretch of DNA. I went over how PCR is done in a previous lecture, so I won't do it again now. But also wanted to mention that PCR is useful for amplifying cDNA as well, because you can do the first steps in a test tube, which is to create cDNA from mRNA, right? You can do that in a test tube, and then use this cDNA as your template for a PCR reaction, and thereby clone many, many, many copies of it. So it's useful that way too. It's really hard to overemphasize how important PCR is. It's used in research, it's used in forensics to distinguish one person from another. It's used in diagnostics, for example, detecting the presence of viral DNA in a potentially infected patient, or to track epidemics that are caused by infectious diseases. It's used to test food for the presence of pathogens, and even to verify the authenticity of foods, for example, whether meat being sold is really what it claims to be, things like that. Alright, most of the rest of chapter 10 talks about how Sanger sequencing works, and we talked about that in the last class, so I'm not going to address that again. If you're not clear on how DNA sequencing works, I would just suggest to reread that section of the book, and of course ask me questions if you have any questions. So finally, I wanted to mention something about reverse genetics. Traditional genetics means that you have a mutant, and it has a mutant phenotype, and you don't know what the gene is, but if you do enough work to isolate the gene, you eventually find the gene that caused your mutant phenotype. And that's how the function of a lot of genes has been discovered. It's how genetics worked for a really long time. But in the times of recombinant DNA technology, you can do something called reverse genetics, which is make the mutant first and then ask what is the phenotype. How is that done? One way in which it's done is called RNAi, which stands for RNA interference. And to tell you about how this works, I need to go over the molecular mechanism first, and I'll first tell you how that was discovered. Researchers that were working with C. elegans, which is a tiny, tiny worm, were interested in blocking translation of certain mRNAs so they could kind of create a mutant that way. And what they thought they would do is give cells complementary RNA that would form double-stranded RNA and hopefully shut down translation of the RNA. They noticed that in some of their worms they were getting an extremely successful reduction in expression of certain genes. And it turns out that what was happening in their worms is the following. Double-stranded RNA is recognized by an enzyme called dicer. And dicer cleaves it into about 20 base pair long fragments. These fragments are recognized by proteins called RISC, and they form a complex where there's a single-stranded RNA called small inhibitory RNA. And that RISC complex will find RNA in the cell. I don't know why this figure calls it foreign RNA, because it could certainly be endogenous RNA. But anyway, it will recognize it and base pair with it and degrade it. It will cleave within this complementary sequence, and the RNA is degraded and destroyed. But the single-stranded RNA in the RISC complex is still fine, and you can go and cycle through this again and again. So this can be exploited. Oh, by the way, before I move on, you may be asking yourself, why does the cell have these proteins? Why does it have dicer? Why does it have RISC proteins? Why does it have a mechanism for degrading double-stranded RNA? Because that's not normally found in cells. That's true. It's thought that maybe this evolved as a way of combating retroviruses. And once the cellular machinery was there, it started to be used for actually regulating gene expression in living things. So it is part of the normal mechanism of controlling gene expression in living things, but it's also used for gene silencing in experiments. And the way that would be done is the following. You would create a piece of recombinant DNA that would, when transcribed, would have RNA of a certain sequence, and then it would loop out, and then the complementary sequence would base pair, and you would have essentially double-stranded RNA. That would be recognized by the dicer and RISC complexes in the cell, and then any RNA that's complementary to those would be degraded. And this is the way to specifically silence expression of certain genes. So if you're interested in gene X, what do? You take a piece of its sequence and create a recombinant DNA construct that will, when expressed in the cell, create some double-stranded RNA that's matching up to gene X's sequence, and then gene X will be degraded in its mRNA. So this is called post-transcriptional gene silencing, because gene X is still being transcribed in your cell, but it's not being translated, and the reason it's not being translated is because RNA is being degraded by the RISC complex. Okay, so this is commonly done in plants. It's also done in C. elegans and some other animals as well. But it's definitely a useful way to make a knockout of a gene of interest and to do reverse genetics that way. Another way reverse genetics is done is through creating a knockout mouse. This is a somewhat complicated process, but not too difficult to understand if we just take it piece by piece. So let's say you want to create a mutant mouse in gene X, whatever gene you're interested in. You have its sequence. Maybe you want to make a mouse model of a human disease, or study development of a mouse that's missing gene X. You start with some embryonic stem cells that are growing in culture, and then introduce recombinant DNA to them. And in some small fraction of those, there will be a homologous recombination event where the recombinant DNA will be introduced into the chromosome. And when it is, it's disrupting the gene that it's introduced into. And that gene becomes mutated, or non-functional. This is a pretty rare event, and you have to use drug selection to select for the ones that have undergone homologous recombination. But you can do that, and then once you've got those recombinant embryonic stem cells in hand, they are introduced into a mouse embryo, and the mouse embryo will incorporate them and be introduced into a mouse, and then grow into a fetus that's eventually born. And then the mice that are born are chimeric because they are a patchwork of recombinant and not recombinant cells, right? And back in the embryo, some of them were from the altered stem cells, and some of them are just normal embryo cells. Anyway, some of these chimeric mice will have germline cells that contain the altered gene. And when those are mated with a normal mouse, you'll get heterozygous mice that are altered in one copy of the gene of interest. And when those are mated together, you can eventually get a mouse that's homozygous, where both copies of the gene of interest are knocked out. So this is a way in which animal models for human diseases are often made. A lot of times the mice are dead. If it's a tricky procedure, because if you're studying a gene that's interesting, sometimes it's interesting because it's necessary for life. And those mice are dead. It's hard to study a dead mouse, or it's hard to study a mouse that doesn't even develop. So this is useful for genes that are maybe interesting but not essential, where you can still study the mutant phenotype. Anyway, these are the main points that are, I think, important for Chapter 10. I certainly didn't talk about everything, but I talked about things that I think will be useful to know, that you'll hear about and that will come up again. And any questions that I would ask you on the quiz would be related to the figures that I've shown here.